Hello, I'm Professor Matthew Schmidt, and welcome to Genetics. We're continuing our series of revisiting Mendel, again in the sense of wondering when we see aberrant results, or at least those that seem odd, does this mean that there's a problem with Mendel's laws, or how are we going to interpret this so that they're consonant with his laws and explain the apparent discrepancy? So this session is a little shorter than some others. It really addresses the phenomenon of lethal alleles, and I wanted to put the idea of pleiotropy in there as well. So let's see what this is all about. It turns out that we're going to see the definition of a lethal allele, but I want to explain to you the experiment that led to the discovery of this. In mice, there are two alleles that exist at one of the loci that controls coat color. Now, coat color in mice is actually a little more complicated than we've let on, but this is the same locus that we were talking about in a previous video that had to do with being agouti versus black, etc. So an allele was discovered that was actually dominant to agouti, and they called it capital A, capital Y to give the idea that it was dominant. Now, it caused a yellow coat color, at least this is the preliminary story, and it turned out that if they took two yellow mice every time, no matter what, two yellow mice, crossed them together, what you see is a strange 2-to-1 ratio of yellow to non-yellow. It turns out that the non-yellow is agouti colored. But a 2-to-1 ratio, guys, it's certainly not 3-to-1. There's no way to make, which is why I put this separate, there's no way to think of a 2-to-1 ratio in the context of 9-to-3 to 3-to-1. It really just doesn't work. No way I can see. So what is the explanation? Well, it turns out that all yellow mice are heterozygous, meaning they have one capital A, capital Y, and one regular capital A. You would think under normal circumstances that a yellow mouse could be capital AY, but we're going to see that that's not possible. But first, let's look at the experiment. So the idea was, after doing some preliminary stuff, I mean, it's sort of a little backwards the way we're thinking about it, but the fact is all yellow mice are heterozygous, but how did we determine that? So if they took a pure breeding agouti line like this and crossed it with the yellow mice, always what they saw was that you got 50% yellow and 50% agouti. We know that this parent can only contribute the big A allele, right? So what that means is the other parent would have to be contributing the big A Y half of the time and big A the other half of the time. So every yellow mouse that was in existence, when they crossed it like this, they always got the 50-50 ratio, which implies strongly that the yellow mouse is in fact heterozygous for this AY mutation and the regular agouti allele. So what is going on here if that's the case? Well, if you do the cross yellow times yellow, so now we know what that means, that, sorry, thought I had my highlighter. Each parent can donate a Y and A, right? So let's follow that. A Y and A Y and A. And let's just fill in the boxes. So we get one homozygous dominant. We get two that are heterozygotes for AY and A. And we get one that's capital A, capital A. So this one looks agouti. As you can tell, this one and this one look yellow. And what's up with the other one? Well, it turns out this little cross through it means that these type of organisms, while they can be conceived, they die before they even implant. I mean, you never will see them amongst the offspring even being born. So since they never come into being, it's almost like saying, remember the conditional probability we did before? It's almost like saying, out of the living offspring, what ratio do we see? And you can tell right from there that it's 2 yellow to 1 agouti. And usually we would see a 3-to-1 ratio, right? Well, unfortunately, the other one that would make up the third, the homozygote, right, is, you don't even want to say it's dead. It really never came into existence in the first place. So this explains the 2-to-1 ratio. And AY is called a lethal allele because when it's present in 2 copies, it causes death. Now, this also leads to a concept that I want to get out there. Why does this happen anyway? I mean, what the heck does coat color have to do with dying if there's too much of this allele? So it turns out that this AY allele has a dominant effect on coat color, but it turns out that that's almost like a side effect, whatever this does, and it's very complicated. It's not fully understood, but the best way I can tell you is that the gene product, in addition to having an effect on coat color, plays an essential role in basic developmental and metabolic processes. In other words, if you don't have a functioning gene of this nature, you can't live. So when it's mutated to AY, it can't function in that way, in the very fundamental way that's necessary for life. But it can cause an effect on coat color. I know it's not immediately clear why this is the case. So death is going to occur unless you have at least one non-mutated copy. So you can have AY with capital A, but 2 AYs, you're not making a certain product that's necessary for development. Okay? So that explains both lethality, it explains the 2-to-1 ratio. Can there ever be a pure breeding line of yellow mice? The answer is no. Why not? That no sort of ended up in a strange place where I didn't want it. But all yellow mice are heterozygous. Heterozygotes are not pure breeding. We just saw if you cross 2 yellow mice by each other, you get the 2-to-1 ratio, right? That's a great question. Now, furthermore, this is an example of a phenomenon called pleiotropy. And pleiotropy means, I'll give you the definition and then we can talk for just a minute about it. A single gene plays a role in determining or contributing to more than one trait or characteristic. It's almost the opposite of something we saw before. Before, we have seen a situation where 2 different genes, 2 non-allelic genes or 2 different loci, come together to determine one trait, right? Here we have one locus that's having an effect, in this case, on 2 traits, and sometimes it can even be more. So what I mean is this AY allele has both an effect on the coat color, and it has an effect on whether you're able to live or die for some other fundamental reason. So this is how I wanted to get pleiotropy in there, but as a general term, it means when you have one gene that's having multiple effects on different traits around an organism. So that's rather a quick one, but I hope you can see why it's still in the vein of these ratios that don't seem to look quite right. And in every case, we're able to figure out, yes, this really was a 3-to-1 ratio. It's just that one of the unfortunate mice in that 3-to-1 ratio never came into existence, so we now see a 2-to-1 ratio. You can always fit it into Mendel's rules one way or another. I'll see you again at another genetics session real soon.